Okay, we're talking about collagen defects and disorders. I will try and make this quick and as pain-free as possible, because it's quite a boring topic. Okay, so just as a recap, there's kind of four main types of collagen. Type 1 is used for bone. Notice how I've written that. It's made by osteoblasts. Type 2 makes cartilage, made by the chondroblasts. Type 3 makes the arteries, and type 4 makes the basement floor. Okay, so there's 4 stages of collagen synthesis. Stuff can go wrong in each of them, and there's some kind of less relevant information as well, which I put in smaller text italics. Our first stage is our cell is going to secrete 3 alpha chains, and these 3 alpha chains are rich in the amino acids glycine and proline, and they're held together by disulfide bridges. These disulfide bridges kind of help the chains form into a triple helix. So this triple helix is called procollagen. Okay, now step 1.5. It's not actually called 1.5, so don't quote that it's called 1.5, but it happens between 1 and 2, so I just called it 1.5. Okay, so there's an enzyme called prolyl hydroxylase. Now what it's going to do is it's going to react with vitamin C and convert the proline in these chains into hydroxyproline. So it's going to add an OH group. Now it's going to happen to each of these chains in the triple helix. So this one, this one, this one. But just for simplicity, I'm just showing it as one chain. So it's going to add vitamin C and prolyl hydroxylase. The vitamin C is going to reduce this basically and add a hydroxy group in it. A less important point again is that vitamin C is an antioxidant, and therefore I think of it like this. So if it's an antioxidant, it's going to take away oxygen because it's antioxidant, and that's how it works. There's an iron atom in here which has oxygen in it, and this vitamin C is going to take away oxygen, i.e. reduce it to form this OH group. Again, a minor point, so don't worry about it. Okay, we've now got this bit here, and you can see it's made up of a green bit. So our hydroxyproline is a green bit with a red useless bit attached to it. So our next stage is getting rid of the red useless bit with an enzyme called procollagen peptidase. It comes along and snips it off, leaving only our nice useful green bit. So our green bit with the OH group, and this is called tropocollagen. Now tropocollagen, so remember this is going to happen to all 3 of the chains, but I'm just showing it as here as one. Now, so we're going to have one helix with the OH group that's been cleaved, and another that's also been cleaved with an OH group. And they're going to come along and happily interact with each other because they both have OH groups. So OH groups form hydrogen bonds, as I'm showing you. So hydrogen bonds between each other, which kind of weakly attracts the triple helices between them, leaving a 40 nanometer gap. So this stage is called assembly of tropocollagen, and it's important to know about the 40 nanometer gap. Our final stage, so remember obviously hydrogen bonds are kind of like a temporary kind of weak form of bonding. Our final stage is making that a nice strong bond, and that is via the enzyme lysyl oxidase, and these are going to cross-link him, covalently link them. So aldi and lidol, the cousins, they're going to have a baby with the enzyme lysyl oxidase, and their baby is called aldol. So lysyl oxidase forms the aldol link between the triple helixes, which kind of keeps them nice and covalently bonded, and the aldol links are between the lysine and the hydroxylysine groups. I don't know what they are, but just learn them. Okay, right, so now on to diseases. There's basically diseases at every stage. The first one we're talking about is one called Osteogenesis Imperfecta, which affects stage 1. So remember, stage 1 is the secretion of the procollagen triple helix. In Osteogenesis Imperfecta, this triple helix can actually unfold, which means the tropocollagen is not going to pack nicely because it's all over the place, meaning our collagen is actually going to end up quite weak. And so you're going to get kids who present often with frequent fractures, for example, especially of the elbow. There's 2 types of osteogenesis imperfecta, type 1 and type 2. Type 2, you never will see a live patient having it because it's so severe it kills the baby. So type 1 is the one again that we see. It's dominant inheritance. It may cause hearing loss, deformity, impaired tooth development, impaired stature, so being shorter. You kind of have a blue tinge on your eye, and an increased risk of fractures, especially of the elbow. So you may kind of see something like this. You have the elbow here, and you can see the fracture here of the olecranon. Here you can kind of see a kind of bluish grey tinge of the sclera of the eye. So again, this is seen in osteogenesis type 1. Again, we said type 2. It's more severe, but that also means that it's rarer, because more severe things are rarer. So this is recessive because you need 2 copies of it. And histologically, you see, this is the trabecula of the bone. That's the main part of the bone. This is apparently smaller than normal, and our weight, which is the gap, the marrow space, is bigger than normal. So our bone is actually weaker. And that's what we can see in type 2. Right, onto the genetics of osteogenesis imperfecta. There's 2 kind of genetic things that you need to know. Both of these are separate things. The first one is, so we're talking about type 1, because type 2 you never really see. So type 1 has dominant inheritance, but it may also have something called the dominant negative effect. The dominant negative effect, the analogy is like when one kid has a tantrum, but it's just that one kid having a tantrum is going to ruin the whole party. So what we can have is, all it takes is basically kind of one cell to be mutated, and then that can ruin everything. So we're going to have a point mutation, which is a mutation in a specific part of the DNA strand. And this can often occur spontaneously as well, within the gametes. And so in the collagen A1 and collagen A2 genes, you get a point mutation, and this is going to change. Remember, the procollagen is rich in glycine. This is going to change that glycine into a cysteine, and this one different allele in these one gene, or these two genes, is going to wreak havoc on all the other genes of collagen, because this one negative gene is going to come along and basically ruin everything. It's going to affect the way that all the collagen sticks together. So all it takes is one naughty collagen to have a naughty amino acid, and then the whole thing is not going to fit together nicely. So that's called the dominant negative effect, and remember this analogy. The other thing that we may see, so this is separate to the dominant negative effect, is germline mosaicism. So this is an exception to Mendelian inheritance, where you kind of get it kind of follows those predictable patterns. This occurs when both of your parents are healthy and they have a gamete cell, so like either a sperm or an egg, and in that gamete cell, one of your gametes is going to randomly mutate. So now, for example, say he's got lots of sperm cells. Say a few of those sperm cells have mutated to cause osteogenesis imperfecta. Again, the majority of the sperm cells will kind of be normal, but there'll be some that are naughty. So what we end up is with a pedigree like this. We have a male and a female here. Both of these are unaffected, as we said. And say, for example, the dad's sperm mutates, but not all of it is mutated. So this is why it's germline, because it's only some of the germ cells. We're going to end up with siblings, and some of them will have the disease, but some of them will not. So that's kind of like a mosaic. We have a different offspring have different phenotypes. So we can see in this case that two have the phenotype and one doesn't, because he's inherited the healthy sperm from the dad and healthy egg from the mom, whereas this son here has inherited a bad sperm, for example, and like a normal as well, because it's dominant. So learn that pedigree, or kind of what the pedigree should look like. So Osteogenesis imperfecta, remember, we said type 1 is the main one, so that's how I remember type 1, stage 1 of this. Our next bit, we're going to skip 2 because 3 is easy to remember, is scurvy. Scurvy has a C in it. C is the third letter of the alphabet. So remember, basically scurvy is when you have a lack of vitamin C. Vitamin C is required for this stage here, but the vitamin C basically adds the OH group. So without vitamin C, we're not going to have the OH group. Therefore, this bit here of forming the hydrogen bonds is not going to happen. Obviously, you'll still be able to cleave it, so stage 2 can happen, but stage 3 will not be able to happen, so we'll not be able to form H bonds. Therefore, obviously, this won't happen either, and therefore, collagen doesn't form properly, and you get hemorrhages, like these little blood spots, and bruising along the skin. So that's scurvy. And our final one is Ehlers-Danlos. Ehlers-Danlos syndrome is called type 7, because there's lots of types of Ehlers-Danlos, but yeah, that's again not so important. What happens here is you have a reduced amount of the scissors enzyme, so a reduced amount of the enzyme that cuts away the red thing, which means the tropocollagen is not properly formed, because we still get tropocollagen with the red, which means that these 40 nanometer gaps will still have the uncleaved bits in it. So as you can see now, because there's lack of this enzyme, there is now all this excess red stuff which hasn't been cleaved, and that is blocking the 40 nanometer gaps. If we block the 14 nanometer gaps, then we're not going to leave room for our enzyme lysyl oxidase to make an aldol link between them. So therefore, we're not really going to get our our collagen is not going to be strongly cross-linked, because our gaps are filled. The gaps that should have the aldol link in them have this useless kind of that's not cleaved getting in the way. This leads to very stretchy skin and hypermobile joints. So you can kind of very flexible, and also a short stature. There's type 4, the illness in type 4 is called Lathering, but I read that mainly affects sheep, so yeah. Alright.